I'm Ann Knowlton, and I'm from the University of California, Davis, and I work on heat shock proteins and inflammation, and the presentation that I did at this meeting was on innate immunity and how it interacts with heat shock protein 60. And what I presented was an accumulation of a number of years of research where we identified that the heat shock response, which is highly protective and it helps remove degraded and denatured proteins, is inhibited in heart failure. And there's no increase in expression of any of the heat shock proteins except for heat shock protein 60, which is an anomaly. And HSP60 is a mitochondrial protein, though about 25% of it is found in the cytosol of myocytes. So we investigated why HSP60 was increased and also whether or not there was abnormal distribution of this increased protein. And what we had found out was that NF-kappa B drives the increase in HSP60, and NF-kappa B, of course, is activated in heart failure. And TNF, which is increased in heart failure, will also contribute to activation of NF-kappa B. And that HSP60 was not only present in the mitochondria and the cytosol where it belongs, but was now found in the plasma membrane fraction. And in further studies, we have shown that not only is it in the plasma membrane fraction, but it's on the surface of the cardiac myocyte. And when it is on the surface of the cardiac myocyte in heart failure from a high LAD ligation in the rat, it is associated with increased apoptosis as evident by activation of caspase 3 and 7, as well as caspase 8. So we were interested in what was the meaning of having this protein outside the cell on the cell membrane, and whether HSP60 might contribute to the progression of heart failure. And so we identified that HSP60 is a ligand of toll-like receptor 4, and we showed that if you administer low-end toxin HSP60, it causes, this is a recombinant human form, it causes apoptosis by a number of different endpoints, including DNA fragmentation and caspase 3 activation. And we can show that if we add a blocking antibody to toll-like receptor 4 or an antibody to HSP60, that we will block this response. And we block the response at several other downstream points from toll-like receptor 4 signaling. The interesting thing was that it wasn't the activation of toll-like receptor 4, it's not known to contribute to the development of apoptosis, but what happens is downstream is activation of NF-kappa B. It leads to increase in TNF expression. The TNF is released, and it's the TNF that causes the apoptosis. And if I add a sequestering antibody for TNF, I prevent the apoptosis. So HSP60 can increase expression of TNF, and TNF can increase expression of HSP60. So we have the potential for a vicious cycle of one enhancing the other and both causing damage. However, we have yet to figure out how HSP60 might get out of the cell, and that is a question that we're continuing to investigate. So it's important to understand that toll-like receptor 4 has a lot of importance in cardiovascular disease. If you inhibit toll-like receptor 4 or mutate it, you can reduce infarct size. And we also cited work, very interesting work from Li Lin in Shanghai, showing that HSP60 released in either isolated cardiac myocytes subjected to simulated hypoxia and reoxygenation, or in vivo ischemia in the rat, that HSP60 activates toll-like receptor 4, leading to more cell damage and to increased expression of TLR2 and TLR4, and that if you add antibody to the HSP60, you can reduce damage both in the in vivo infarct setting and in the culture dish with the isolated rat cardiac myocytes. Now this is all interesting, but there's a lot of data about HSP60 being in the serum of people, and the Whitehall study samples were analyzed, and 1 microgram per mil was found in 25%, while 25% had none and the rest were in between. So there's something we don't understand, and one possibility is that either there is a post-translational modification that makes it inactive or unable to bind, and a second possibility is that it's in exosomes. We have found that HSP60 is released from cardiac myocytes in exosomes, and that the exosomes are remarkably stable no matter what kind of pathological intervention we tried, HSP60 remained inside the exosomes. And people also have antibodies to HSP60, and so there's many possibilities for HSP60 setting up a disease cycle through activation of mostly innate immunity. Because if HSP60 interacts with an antibody in the serum, that will activate complement. You can also activate macrophages, and macrophages are much smaller than myocytes, so anything they release to engulf the myocyte will just go into the environment. The last thing we did is we looked at TLR4 in the mouse, and we confirmed our same findings that we had in rat cardiac myocytes, that if we had a mutant mouse, the HEJ mouse, that has a mutant receptor, we saw that we were able to block apoptosis. However, the surprising thing was that we found that we had in the wild types, when we give HSP60, we have a decrease in viability. Now, maybe that is from an increase because they just have increased apoptosis. However, that if we added, blocked the toll-like receptors or blocked HSP60, we were able to improve viability. So we went on to investigate this, and the most interesting new finding is that it is mediated by activation of PKC alpha if you do hypoxia and reoxygenation within 10 minutes of reoxygenation, followed by an increase in NOX2 expression at 1 hour. NOX2 will lead to the generation of a lot of ROS, which in turn will, of course, injure the myocyte and can cause necrosis or contribute to necrosis. So much work needs to be done, particularly understanding how HSP60 gets out of the cell and whether or not it is truly increased, because there's in heart failure serum, because there's a lot of controversy in this area. And that's work that's ongoing in my lab and in other labs in the country. This is a very good Congress, and I've been a number of years, and every year it gets better. And I feel that one nice thing is there's a greater diversity of speakers than you have at many of the congresses, where you may hear the same people again and again. And there's also more of an international flavor. And the sessions are different every year, and you can attend. I'm also a cardiologist, so I attend the clinically relevant for me sessions that look of interest, and at the same time I can hear some interesting basic science, as I did yesterday in the session that I chaired. So I think it has a nice combination, that the people who come are very good, that like this year we had for the I guess it was the Nanette Wenger's presentation. I mean, we just every year we have really star people come and provide interesting insights and an overview based on many years of their experience in cardiovascular research. So I think it's a rich environment, somewhat different from other conferences, and I find it very rewarding. I try to always have it on my calendar.